Our eyes are one of the most magnificent sensory organs known in nature. They allow us to detect light with a dynamic range that spans 10 orders of magnitude. On one end of that spectrum, we have the ability to see under very bright conditions, such as at the beach during the summer, but we can also see very dimly lit stars at night. And so what underlies the remarkable ability of the eye to see over this large dynamic range? This is one of the key questions that we address in my laboratory here at UCI. We use a variety of techniques, including in vivo mouse models, biochemistry, enzymology, and structural biology to address this question. What I'd like to share with you in this presentation are three interrelated stories where we've used structural biology to investigate the function of a protein that plays a key role in maintaining the light responsiveness of the eye. Vision begins whenever light is focused into the back of the eye onto this paper-thin tissue known as the retina, which contains cells known as photoreceptors, which are responsible for detecting light. These cells are able to detect light because they express proteins called visual pigments, which consist of a protein component opsin and a covalently bound chromophore 11-cis retinal. Whenever the 11-cis retinal absorbs light, it undergoes a configurational change from cis to trans, which results in conformational changes in the opsin protein, allowing it to initiate the signal transduction pathway. After this process occurs, the visual pigment releases its chromophore and is insensitive to further light stimulation. So therefore, there must be biochemical pathways in the eye that regenerate 11-cis retinal for opsins. These are known as visual cycles. There's a number of steps and a number of proteins involved in the visual cycles, and my laboratory is interested in most of these. However, for this presentation, I'm going to focus on one particular protein called RP65, which carries out a hallmark step in converting an all-trans retinoid into an 11-cis retinoid. The reaction catalyzed by RP65 is unusual from a biochemical point of view because it involves actually two half reactions, an atypical ester bond hydrolysis and an alkene isomerization from trans to cis. This is thought to occur via carbocation intermediate, and it's known that RP65 depends on iron for this activity. The approach we've taken to understand how RP65 achieves this catalytic mechanism is to use mimetics of retinoids for structural studies. In particular, we focus on a molecule called a mixostat, which has retinoid-like features, but is much more water-soluble and amenable for structural studies. This approach has been successful. We were able to determine the structure of RP65 in complex with a mixostat and also the palmitate product of catalysis, where we observed that this palmitate is coordinated to the iron. This provided two key insights, one of which being the orientation of the substrate during catalysis, but also provided insights into the role of iron in catalysis. Namely, we proposed that iron is functioning as a Lewis acid to polarize this ester and facilitate its dissociation during a reaction. Looking at the RP65 active site in more detail, we were able to identify particular residues that seem to allow the stereoselective 11-cis isomerization reaction to occur. Namely, these are residues phenylalanine 103 and threonine 147, which form electrostatic interactions at a key position to stabilize the carbocation intermediate at the C11 position. RP65 belongs to a large family of enzymes known as carotenoid cleavage dioxygenases that typically cleave alkene bonds within substrates. As an example, I show here a phylogenetic tree with RP65 embedded with a set of proteins called BCOs, BCO1 and BCO2. To give a specific example, BCO1 takes beta-carotene and using molecular oxygen cleave it to form two molecules of retinal. So this raises two important questions. One, how do these alkene cleaving enzymes such as BCO1 choose which of these chemically similar alkenes to cleave? Okay, so it's regioselectivity. The other is, what is the relationship between the catalytic activity of RP65, which is an isomerohydrolase? How does that relate to the alkene cleavage activity of these traditional carotenoid cleaving enzymes? So again, we've used structural biology to address this question, and the particular CCD we've had recent success with is one from archaea, which we were able to crystallize in complex with its apocarotenoid product, as shown here. By examining the active site in complex with this apocarotenoid product, we've been able to determine the key features of the active site that allow the positioning of the correct alkene bond over the iron for reaction with dioxygen. Another important insight that we obtained from this study was the structural differences and similarities between this alkene cleaving enzyme and RP65. We can see in both active sites that there's a region of negative electrostatic potential, which we believe could help stabilize a common carbocation intermediate between these two enzymes and their catalytic mechanisms. But what you can notice is this difference in the active site geometry when we compare the alkene cleaving enzyme to RP65. Specifically in RP65, the active site adopts a bent conformation, which we believe would help facilitate the transition of a trans retinoid into a bent cis configuration. Now, as I mentioned, our vision enables us to see over a very wide range of illuminations, and it happens so seamlessly that it's easy to take it for granted. But actually, proteins involved in maintaining the sensitivity of the eye to light are affected in certain diseases. In particular, the one that I'll talk about today is retinitis pigmentosa. For example, mutations in RP65 can cause a blockade in the visual cycle metabolic pathway so that we can't form 11-cis retinal for opsins, and that results in this disease, retinitis pigmentosa. There have been a number of mutations in RP65 that have been identified as causative of retinitis pigmentosa. But the one I'll focus on for this presentation is a newly discovered mutation that leads to a dominant acting form of retinitis pigmentosa. This is the first dominant acting RP65 mutation identified. This occurs, this mutation is in an aspartic acid at position 144, converting it to a glycine. It's of interest because not only because of its unique clinical features, but also because of the location of this position within the RP65 structure. If you take a look, this aspartic acid is located out on a loop far away from critical structural elements of RP65, such as the iron center and the membrane binding surface. So it was a mystery why this aspartic acid to glycine mutation would be pathogenic. So our approach has been to use structural biology to investigate the structural consequences of this mutation on the RP65 structure. Now, we couldn't use RP65 directly for these mutagenesis studies because we obtained the protein that we can use for structural studies from native source. So the way we circumvented this was to use an RP65 homolog and generate a chimera in which this protein has been turned into RP65 in this region of interest around this residue 477. So what we found by making the D477G substitution, the structure of the enzyme is not impacted, the tertiary structure is not impacted. However, what we found is by making that substitution, we turn a loop that's normally not prone to forming protein-protein contacts into one that's very, very highly prone to making protein-protein contacts. We had two crystal forms where what we observed that just by making this single point mutation, we turn this loop into a sticky surface that likes to engage in diverse protein-protein interactions. This leads us to our hypothesis that by getting rid of this negatively charged residue in this region, we turn a normally non-sticky surface into a sticky surface. So in effect, we've generated a neomorphic surface with potential, so it's in fact a gain of function potential to engage in abnormal protein-protein interactions. And we believe it's these abnormal protein-protein interactions that lead to the unique clinical features of this form of retinitis pigmentosa. I hope this short presentation has given you a flavor of the types of questions we're trying to address related to the physiology and pathophysiology of vision, as well as the approaches that we take to address those questions. If you have further interest in our research, please don't hesitate to contact us.